I'm Stephen Taylor and I'm head of the Respiratory Health Group at the South Australian Health and Medical Research Institute, and we conduct research across a range of different projects to understand chronic lung diseases to really try and improve the way that we can deliver effective treatments. So I actually came from a mucosal immunology background, and that was when I graduated from my undergraduate degree in 2013 up in Brisbane. And I conducted my honours looking at how respiratory viruses can actually infiltrate into the epithelial cells of the lung. It was only after a year of doing that honours that I really kind of had a passion for research and wanted to sort of understand more about how the host, so how our lung interacts with different microbes. So after a year as a research assistant up in Brisbane, where I was collecting respiratory samples as part of a partnership with a respiratory physician, I decided to conduct my PhD down in Adelaide at the South Australian Health and Medical Research Institute to understand more of the microbe side, so understand how different bacteria and viruses both colonise and cause infection in the lungs. And so I conducted my PhD at SAHMRI under the guidance of Professor Geraint Rogers, and what we did was we characterised the respiratory microbiome across a range of chronic lung diseases. And from there I really got an appreciation for the complexity of the interactions between the host and the bacterial populations, and also how bugs can both be associated with causing disease, but also they can be protective, or they can be associated with less severe disease. And really understanding those interactions has been what kind of keeps me up at night and keeps me thinking about the respiratory microbiome and how we can better apply that in the clinical practice. I conduct research across a wide range of chronic lung diseases, so they can be as common as childhood asthma, which is, I guess, the most common respiratory disease, through to quite severe, more rare diseases, including cystic fibrosis or primary ciliary dyskinesia. And across that whole spectrum, I really try and apply different molecular tools to understand the important clinical questions depending on the context. So as an example, in childhood asthma, what we know is that it's common across the world, but a lot of the research that's actually conducted has been done in more metropolitan, high-income countries, where we have characterised the different interactions between the immune system, the respiratory tissue, the epithelial cells, and the microbes to a high degree. But what we really don't know is whether we can I guess, expand that knowledge or that insight to other areas, so particularly more rural areas or areas of low to middle income countries. So some of the research that I've been doing has been really to try and expand our understanding and looking at more low to middle income countries, and to be able to characterise the immune system, the airway microbiota, so the bacteria in the lungs, and see whether those interactions hold true across childhood asthma globally. Another area that I'm quite passionate about is those people with quite severe chronic lung diseases, and that includes cystic fibrosis or bronchiectasis, where what happens is they get colonised by really nasty pathogens, including non-tuberculous mycobacterium or Pseudomonas aeruginosa. And these bacteria can be really antimicrobial resistant. They can be very difficult to treat. And what we see is treatment failure often occurs. So I sort of apply different, again, molecular techniques to try and understand why might treatment be failing. So that might be due to antimicrobial resistance, or what is the other microbes in the lung environment of those patients, and might they be playing a role in either treatment success or treatment failure. So by characterising the respiratory environment in these different contexts, I can really start to piece together which microbial ecosystems are more I guess, beneficial in terms of treatment outcomes, and which ones might be things that we need to really kind of focus on and give us a prospective insight into whether a treatment might work or not work. I really find it interesting trying to understand what is going on in the lungs. The lungs are incredibly complex. It's a very distinct and intricate architecture of bronchi, bronchioles, alveoli, and what we see in chronic lung disease is the deterioration of this lung tissue, promoting a really complex environment that promotes a bacterial colonization, and it's nearly a hotbed of quite stochastic or maybe even predictable interactions between the host and the microbiome. So I'm really kind of passionate about just trying to tease that apart, and it's really interesting because every time we look at a sample we get a new bit of insight into what interactions are happening in that particular individual, and maybe at a whole cohort level we can start to see patterns that emerge that we can then apply to be able to better understand a new person that develops a chronic lung infection. Outside of the lab and outside of the office where I do some bioinformatics, I really enjoy just kind of spending time outside, especially with my rescue dog and my wife. So we go for long walks, we go camping. He's a staffy, so he doesn't have the greatest endurance. He likes to go for a really quick run and then kind of fall asleep. So we like to just sort of take him on various different adventures. I also like to go cycling. Adelaide is very flat, very prone to, or very helpful for cyclists that maybe don't have all the endurance of the people that do things like the Tour de France, but it's very, very fun.